U of T is very strong in the traditional engineering disciplines, like computer engineering or mechanical engineering, and those are areas that my research has roots into. So that was a very attractive thing. The other thing is IBBME, the Institute of Biomaterials and Biomedical Engineering, has this perfect blend of faculty with a lot of expertise, a lot of experience that have generated a very solid base of knowledge. And then this very young faculty with very innovative approaches. Not that the older faculty are not innovative, but this perfect combination of solid, experienced faculty and younger faculty. So that was the main attraction to IBBME. And finally, Toronto is a big hub for cell biology and developmental biology, and a lot of my work is into those two areas. So I thought this was the perfect combination between engineering and biology, where I could find collaborators in both areas. What I look forward to the most is interacting with the students, so both in research and in teaching. And my wish is to stay at the bench as long as possible, and to do experiments myself. But we all know realistically the people who do the science these days are people who are at the bench, and this is mostly students and postdocs. So I really look forward to the interactions with these people, especially the people who join my lab, and to learning from them, because I think that's the beauty of science, right? That the people who interact with you every day teach you the most. So what we are interested in is how embryonic tissues heal wounds, and in particular we focus on the epidermis because, well, it's very accessible and it has human relevance, of course. Now, it has been demonstrated in many different organisms that the epidermis in embryos heals wounds much faster, much more efficiently than the epidermis in adults. So if we can understand the mechanisms that drive wound closure in the embryonic epidermis, we may be able to then engineer strategies to accelerate or improve the wound healing response in adults. Now you're probably wondering, how does engineering fit into this problem, which sounds very biological in nature? And the way it fits in is that people have generally looked at wounds and assessed qualitatively, how is this wound closing. But we bring in the ability to image the wounds live as they heal, because we do this, and I haven't mentioned that, in fruit fly embryos. And the wound healing response in embryonic tissues is conserved. It happens very similarly in the fruit fly embryo, in the mouse embryo, or in the human embryo. So we can take the fruit fly embryo, put it under the microscope, and watch how wounds close live. Now, this generates huge amounts of data that we need to then process and analyze, and we do this using image analysis algorithms. And I guess the other aspect where engineering comes in is that I'm very interested in how the imbalance in mechanical forces that the wound generates in the tissue affects the wound healing response. So the idea is that perhaps these changes in the balance of forces may be triggering the wound healing response itself. So that's another area where engineering will come in to try to manipulate the forces that cells experience in the living embryo.